Assalamualaikum warahmatullahi wabarakatuh. So today I with my team will present the enzyme linked immunosorbent assay, or we call ELISA. My name is Nurhani Suzwani binti Nur Hizam 23BB08042. My team member Nur Najatul Wafa binti Muhammad Rohaini 23BB08036. Siti Nur Ain Atika binti Suhaimi 23BB08058, Nur Fitrah binti Ahmad Syahir 23BB08059, and Nurul Shazwina binti Rosli 23BB08061. As you can see here, this is the sample of a ELISA technique.
I will continue the presentation by explaining the meaning of ELISA. ELISA is enzyme-linked immunosorbent assay. It's a laboratory technique used to detect and quantify substances such as proteins, antigens, antibodies, and hormones in biological samples such as blood, serum, or urine. ELISA was based on the interaction between an antigen and an antibody, which is linked to an enzyme. This enzyme produces a color change that can be measured using a spectrophotometer. Other than that, ELISA is widely used in veterinary medicine and animal research for diagnosing disease, monitoring immune responses, and detecting pathogens in livestock, pets, and wildlife. There are 4 types of ELISA. The first one is direct ELISA and the second one is indirect ELISA. The first one, direct ELISA, the process is about an antigen is immobilized on a microplate and an enzyme link antibody directly binds to the antigen. The process for direct ELISA is fast and simple for a few steps, and the cons for direct ELISA is a higher background noise and lower specificity. And the uses for direct ELISA is detecting large antigen concentrations, vaccine development. However, for indirect ELISA, the process is about an antigen is immobilized on a plate and a primary antibody binds to it. A secondary enzyme linked antibody then binds to the primary antibody. The pros of the indirect ELISA is higher sensitivity, amplified signal, more flexible detection options. The cons for indirect ELISA is more steps, potential for cross-reactivity. And the uses for indirect ELISA is measuring antibodies in a sample like HIV and COVID-19 testing. The next type of the types of ELISA, the third is competitive ELISA, which is inhibition ELISA. The process for this type is the sample antigen competes with a labeled antigen for binding to an antibody. The stronger the signal, the lower the antigen concentration in the sample. The pros for these types is works well for small molecules, high specificity. The cons for this type is requires well-optimized conditions, more complex. And the uses for these types of ELISA, competitive ELISA, is measuring hormones, toxins, and small molecules, examples like drug testing. The last types of ELISA is sandwich ELISA. Sandwich ELISA is a captured antibody is immobilized on a plate and the target antigen binds to it. A second antibody, which is detection antibody, is added, followed by an enzyme-linked secondary antibody. The pros for this sandwich ELISA is high specificity and sensitivity, and the cons for sandwich ELISA is requires 2 specific antibodies for the target antigen. And the uses for sandwich ELISA is detecting cytokines, tumor markers, and infectious diseases.
Assalamualaikum warahmatullahi wabarakatuh. So for today, I will continue my teammate presenting. So I will explain about 5 main use of ELISA. Okay, for the first point is medical diagnostic. ELISA is widely used in hospitals and laboratories to detect infectious diseases such as HIV, hepatitis, and COVID-19 by identifying specific antibodies or antigens in patient sample. It also helps diagnose autoimmune disease like lupus and rheumatoid arthritis by detecting abnormal immune system activity. In endocrinology, ELISA is used to measure hormone level. For example, insulin for diabetes or thyroid hormones for thyroid disorders. Additionally, it can detect cancer markers such as prostate-specific antigen (PSA) for prostate cancer screening. Okay, for the number 2 point is food safety and quality control. ELISA is crucial in food testing to identify allergens like peanut, gluten, or milk proteins, which can cause severe allergic reaction. It also detects contaminants such as mycotoxin, toxin from fungi, pesticides, and bacterial toxin that can pose health risks. Okay, for the point number 3 is pharmaceutical and drug testing. In medicine, ELISA is used to monitor drug level in patients to ensure the right dosage is given for effective treatment. It is also used in forensic toxicology and law enforcement to screen for illicit drug use, such as detecting substances like cocaine or opioids in blood or urine sample. So, point number 4 is veterinary medicine. Just like in human medicine, ELISA is used to diagnose animal disease, such as foot and mouth disease, FMD, and brucellosis in livestock. It also plays a role in vaccine monitoring, ensuring that animals develop the necessary immune response after vaccination. Okay, for the last point is environmental monitoring. ELISA helps in detecting toxin and pollutant in water, soil, and air, including heavy metal, bacterial toxin, and harmful chemicals. It is also used in biodefense to identify potential biowarfare agents, such as anthrax, ensuring public safety. So, by providing high sensitivity and specificity, ELISA remains a powerful tool across healthcare, food safety, pharmaceutical, veterinary science, and environmental monitoring. So, that's all from me. I will pass to my teammate.
So next, application of ELISA. ELISA serum linked immunosorbent assay is a widely used biochemical technique for detecting and quantifying substances such as protein, antibodies, hormones, and pathogens. Its applications span across various fields including medicine, research, and industry. There are 4 applications of ELISA, which is medical diagnostic, infectious disease and detection. ELISA is used to diagnose diseases like HIV, hepatitis B and C, tuberculosis, and COVID-19 by detecting specific antibodies or antigen. Next, pharmaceutical and drug testing. Drug monitoring used to measure drug level in patients undergoing treatment, and vaccine development. Help access to immune response to vaccine by detecting antibody production. And third, food industry safety, detection of food allergens, identify allergen like gluten, milk protein and nut in food product. Other than that, pathogen detection used to screen for bacteria such as Salmonella and E. coli in food and water. And finally, research and biotechnology, protein and antibody quantification used in lab to study protein interaction, immune response and molecular pathway.
So the next slide is advantage and disadvantage ELISA. Advantage and disadvantage based on the type of ELISA, which is direct, indirect, competitive and sandwich. First one, direct. Advantage of direct is rapid and easy setup, simple procedure. Disadvantage of direct is less specific since it employs single antibody detection. Antibody should be conjugated and potential for high background signal. The second one is indirect. Advantage of indirect is application of signal using a secondary antibody, high sensitivity as compared to direct ELISA, and cost effective. Disadvantage of indirect is longer protocol due to greater number of steps, potential for cross-reactivity caused by secondary antibody leading to non-specific signal. The third one is competitive. Advantage of competitive is ability to quantitate small molecule with low analyzed concentration and sample purification not required prior to use. Disadvantage of competitive is low sensitivity and specificity. And the last one is sandwich. Advantage of sandwich is higher specificity and sensitivity, and antigen purification are not required prior to use. The last one is disadvantage of sandwich is cost and time consuming, require matched antibody pairs, and long procedure.
Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Nora Ainatika binti Suhaimi and my metric number is 23BB08058. Now I will proceed to the next part of the presentation, which is differences between ELISA and other techniques. ELISA, or enzyme-linked immunosorbent assay, is a widely used biochemical technique for detecting and quantifying various substances, such as proteins, hormones, and antibodies. It plays a crucial role in diagnostic and research due to its specificity and sensitivity. However, it differs significantly from several other commonly used techniques in the field of molecular biology and immunology. The first one is differences between ELISA and Western blot. ELISA is a rapid, quantitative, and highly specific method for detecting proteins and antibodies, making it an essential tool in diagnostics. In contrast, Western blot is primarily used to confirm ELISA results, as it not only detects proteins but also provides information about their molecular size and structure. The second one is differences between ELISA and PCR, polymerase chain reaction. PCR is a technique designed to amplify and detect genetic material, such as DNA and RNA. This makes it more suitable for genetic research and infectious disease detection. On the other hand, ELISA is specifically used for detecting proteins and antibodies in biological samples rather than genetic material. The third one is differences between ELISA and immunohistochemistry, IHC. Immunohistochemistry, IHC, is a technique used to visualize proteins within tissue samples, making it particularly valuable in pathology and cancer diagnosis. In contrast, ELISA is performed in liquid samples and provides precise, quantitative data on protein concentration. The fourth one is differences between ELISA and flow cytometry. Flow cytometry is primarily used for analyzing individual cells, making it a powerful tool in immunology and oncology research. Unlike ELISA, which measures the overall amount of a specific protein or antibody in a sample, flow cytometry provides detailed insights into cell populations and their specific characteristics. In the conclusion, in summary, ELISA is a highly efficient, easy to perform, and automatable technique, making it ideal for high throughput analysis in both clinical and research settings. While it shares similarities with other molecular and immunological techniques, each method has its own unique applications and advantages. Thank you.
So the last slide is conclusion. ELISA is a sensitive and versatile technique used to detect and quantify antigens or antibodies. It comes in different types, including direct, indirect, competitive, and sandwich ELISA, and it widely used in diagnostics and research. While ELISA is cost-effective and simple, it has limitations like potential false result compared to other methods. It offers faster results but less detailed information.